I think Chuck Messing thought that he probably wasn't actually swimming, but just getting caught in a little bit of a current.
Yeah, that's what I was wondering, because I've never heard of him swimming.
Yeah, he said that genus doesn't usually swim.
But
we also heard back from Eric Cortez on the coring of mounds in the Gulf of Mexico,
come up
of
a little bit,
Lophelia
like,
mounds.
it drags me back. Then I come down and...
Yeah, so current grounding effect kind of.
He said that 16 meters they drilled into
like,
coral mound, a Lophelia mound in the Gulf of Mexico, and they found nothing but coral skeleton, and at the bottom it was 300,000 years old. Wow.
Right there.
Okay.
The total mound depth that they got from seismic data was 20 meters. So here we are at one that's 3 times that size.
Thanks, Bridge.
Geologic time is very hard to grasp, even for me, and I'm a geologist.
Okay, the thickness of the mound was 20 meters, and the water depth was 400 meters. So we're quite a bit deeper here. But it would be interesting to see what the seismic data would say about the thickness of this mound. Or ridge, or it's definitely different, very different shape than the places we have been the past few days.
A ridged mound. Perhaps it was
it's definitely hard.
A mound shape before, and now it's been eroded into.
I'm gonna rotate you up.
Let's come
There's a very large scour mark at the base of the southwestern facing slope. So perhaps the Gulf Stream shifted its direction and the currents attacked that edge and might have scoured it into its current shape.
Just grab it.
And did that slowly enough so that the corals could still continue to grow. We saw a tremendous amount of rubble, coral rubble at the base of the slope. It was on the lee side of the current, on the sort of northeastern facing side. But we haven't explored the base of the other side facing the current.
Have them come out.
Let's come out video. Sorry, watch lead. I tried to get both.
Thank you, pilot.
Let's keep on moving up.
Get stuff because you can get sort of like a gusting effect too sometimes.
Yeah, probably you won't have as much problems as I am, but yeah, this is the first time I think
And we're up on the ridge of a very steep face slope that we climbed, and now is this sort of, there's an interesting planktonic organism. Is that a...
wonder if that's a siphonophore.
So I was afraid to say it.
I didn't see the whole thing, but oops.
So we're now at the top, and this is different. We're seeing an area that doesn't have a wealth of coral community activity going on.
Going mostly up.
Or the rubble area.
There's a little bit of a dip between.
Yeah, the ridge may be highs and lows, or actually maybe we were just on the edge, the flank of the ridge, instead of at the peak. It's so narrow, it's easy to miss the top of it.
Yeah, it's quite different than where we wound up yesterday, where we had this very broad crest of the mound.
Okay. So
yeah, it really looked like the top of the mound. Put
your head into the current.
Broad and flat.
You could see the edges sloping off in the distance. This very different, very peaked. And all those corals growing upward as much as they can to take advantage of the currents. It's a nice shot illustrating the flow of the water.
Is that move still going, or is that better?
The move is done.
You could probably push me a little.
4 minutes ago already, but you haven't moved a ton
Not
closer.
in the distance. You see a field of
as I come up and down, I'm going to
more colonies.
Back.
Okay.
So
understood. You want me to bring you in a little closer to him? Or wait a minute?
We can sort of split the difference. Yeah.
You want like a 315 or something? Or
sure.
310? Okay.
Yeah.
We'll do 310, 20 meters.
Sounds good.
We're sort of peeking over the edge, looking into the Gulf Stream, looking in a southwesterly view
I'd like to move 20 meters, bearing
all the way down the
310
slope
degrees,
as far as we
speed
can
decimal
see.
2.
Abundances of these Lophelia corals.
Good gobbler.
What was that, Barry?
And this ridge feature has about at least 80 meters vertical relief off the seafloor.
You're off to starboard.
The seafloor was 870 meters, and we're now at 785 meters.
Yeah, it's actually 110. I think is what the estimate was to begin with.
Let's
quite a substantial ridge.
Hold right there.
There's another coral hake.
Move initiated. Thank you.
Lamanema melanurum.
Right there. Hold.
There are a lot of crinoids here.
Partial out.
Alright, you gotta push the port here.
Okay.
You're getting blown off.
Ken Sulak was commenting on the
if 310 is a little bit too far to starboard drive.
The skate that we saw a lot earlier today. I don't know if you were here for the beginning of the dive. We saw a very small patterned skate, modeled pattern skate. And I think that this is the name that Leslie came up with also. Fenestraha plutonia. And he said that the tail is held absolutely straight to avoid distorting the electrical field that's used in electroreception. Oh, and we have another one of these sea stars like we saw earlier.
Adjust it. Okay. Bridge is RV nav. Yeah, can we hold present position, please?
He is closer to live coral than the one that we saw previously, I think.
Yeah, I'm thinking more like 280.
Chuck Messing has identified the feather star crinoid as Xenometra columnaris. Extremely long cirri, or the hooks arranged in columns, and the arms extend in slender filaments as characteristic to this species.
All right, 20 meters, bearing 280 degrees, speed decimal 2.
And he says that this species occurs in shallower water in the western Atlantic in the depth range of 500 to 1,000 meters than it does in the eastern Atlantic, where it occurs between 3,400 and 4,000 meters.
Wow.
That's interesting.
Yes, you don't want to go out too far that way.
All right.
Maybe we just haven't looked that deep for them here.
Yeah.
You want to come back to port.
But that is interesting. And we're right in the depth range that you mentioned, Chuck. Thanks for that information.
Move initiated. Thanks, Bridge.
That's where we're going with the ship, but right now. There you go.
Okay.
Chuck is at Nova Southeastern University. He's been following us on many of these dives.
Playing at the end of the leash.
The Venus flytrap anemone. And some more crinoids. I don't know if that's Madrepora that they're on, but they sure do blend in well.
Let's come in, video. Hold right there.
Go ahead, watch lead. Sure, we can do that.
We'd like to introduce or have our front row introduce themselves, please.
City Nav is Levi Unima.
Piloting right now.
It's Fernando Aragon.
I was going to make the announcement for you, Fernando, because you're concentrating so much.
All right.
Co-pilot is Josh Carlson.
Over on video, Bob Knott.
And back in the back row clipping, Annie White. And we're all with the Global Foundation for Ocean Exploration.
And in the back row,
Okay, let's come in
Derek,
a
would
little
you
bit
like
more
to
video.
introduce yourself?
Hold right there.
So once you're stable,
Yeah.
just go right
Hey
in.
folks,
Try to get
this
a
is
good
Derek
shot.
Sowers.
But
I'm with
I'm
Noah's
never
Office
really
of Ocean Exploration
fully
You're not going to be
Research, not being
stable for
lead.
very long, and you're never going to
Okay.
be fully stable.
Okay.
And this is
I'm
Cheryl
never
Morrison
even
from
fully
the U.S.
stable. So you come
Geological
in as far
Survey
as you
Leetown
can.
Science Center, and
Get
I'm
as stable
the
as you
biology
can.
science
Try to
lead.
get the shot.
Okay.
And I'm Leslie Sauter from the College of Charleston Geology Department.
Like right now, I'd be
And
like,
I'm the
right, go in.
geology lead.
All right.
Okay. All right. Pull out.
Go ahead, video.
And we are currently on our 7th dive. We have crisscrossed the Blake Plateau, and now we're heading out.
Hold right there.
After this dive, we'll do another dive in this general region. Then we go
but
into
I'm
deeper
drifting
water again,
back.
briefly.
Yeah, partial out. Okay, let's just come out, video.
How trippy would it be to have the Zeusonic gimbal?
And we had to run a long way east today to find a place to put the ROV in where the conditions would allow it.
Be like flying
Yeah,
a drone.
it was looking a little sketchy for a dive for a while. But,
Okay.
so they've been gracious to allow us an extra couple of hours. Normally we would be leaving, we would have left the seafloor by now.
And then you're
But they've extended this
you don't get
dive,
to do that,
and we'll be
I mean, you got the
leaving
cams.
the sea bottom
Yeah, I mean,
roughly at
you
6
can
o'clock
fly
Eastern
just
time.
from the minute cams.
So I hope you
I
can
do
stay
most
with
of the
us.
Yeah.
When,
Those two cameras
Maybe
give
have
you a
your
good
hors
perspective on
d'oeuvres
your motion.
Yeah.
for dinner with us. So, Cheryl, maybe you could tell the audience listening about the difference between these deep sea corals and the type of coral that most people are familiar with in the shallow waters, and particularly their coloration. What we see is a lot of white color here, which a lot of people think of as a bad thing. Bleaching of corals, you may hear about in the news.
Right.
Hold right there.
That is a bad thing, and it is happening
in
shallow water corals, not here. Shallow water corals have zooxanthellae, which are algae, which live symbiotically in the tissue of the coral, and it's what gives them their color. And if they're exposed to too much heat for too long, those zooxanthellae beat feet, basically. They leave the coral, and the coral then bleaches or looks white. Sometimes the coral can come back and survive that kind of situation, but sometimes they cannot. These corals that we see down here don't have zooxanthellae. There would be no point in it because there's no sunlight that reaches these depths. So instead, they rely on feeding in the water column. So that's why they like to be up high in the water column and where there's a lot of water flow, which delivers the food that they capture out of the water column. And so
right when you zoom,
they are white, but that's okay. That does not mean that they're bleaching. And there are species like the madrepora that we see that are colored, but yeah, it's not due to zooxanthellae like we see on shallow water reefs.
Yeah, actually, in this region, if you're brown, that's a bad thing because that means the coral has died and the iron and magnesium in the water are starting to precipitate onto the coral structure. So all the brown material is not healthy at all. It's dead.
Right. It's dead, but it's also still creating a habitat for other organisms as well as building up these mounds. So very important structure.
Maybe another sea star.
Oh, yeah.
Same type that we've seen now. This would be the third.
Okay,
if it is in fact the same species, it looks very similar.
So just watch your minute cams.
Yeah, a lot of times I'll like stare at the manips and then in the peripherals try to center
down
them
a little bit. Watch your left tail.
No, but it was rotating. Let's come out partially. Video.
Have a glass sponge back there, and the two of them on the left.
Yeah, I'll take your main,
Okay.
your rotator.
Going video.
Yep, that looks very similar to the one we saw previously.
That fleshy material between the arms.
Go ahead, watch lead. Let's come out. Video. You have me latched in, I think, right there. Okay.
Trying to plan my next move, Josh. Looks to me like Hypax got it.
We're going to zoom in on this
Maybe
crinoid again.
310.
Chuck Messing was hoping to
305,
make
but in real life,
another look, take another
I think
look
it's
at
gonna
it.
put you in
He
deeper
said if
water,
it's the
maybe.
species he believes, that there's only 20 specimens
would
available,
say,
so this
maybe
might be
let him
helpful
acquire
in
ridge
the
and go
ID.
from there.
Yeah, have him push out a little
Okay.
bit after this. And
Yeah.
Video, whenever you're ready. Let's come in
suspect
partially right here.
It's going to be another 20 meters, like 270 or something. But
Hello. Chris Ma.
Hello, Chris. How are
Right there,
you?
hold right there.
So we're going
Good.
after the crinoid.
You
Yeah.
observed yet another Perenniid sea star
This is gonna be
amongst
quite a ride, Annie.
the coral forest.
I have
Yeah.
total faith in
So
you.
I've looked up some of my notes and checked the pictures and double-checked my identification. I think this is something called Chondroaster grandis.
Huh.
And
It's,
unfortunately, so there are lots of records of this from the North Atlantic and things that we've seen sort of farther north.
it's going.
So this is, I'm pretty sure, a
Okay.
range extension. I have to double-check exactly which species this is because the type is from quite a long distance away, and all the specimens in our collection are identified consistently, but I don't know if they're compared against the actual material on which the species is based. So there's some uncertainty, but that's not really anybody's concern but mine until if and when I decide to publish this. But it's definitely a new record, and if we get to see what it's doing, if we get it at another angle and it's feeding on something, that would be an interesting observation. But other perenniids, such as Perennia pulvilis, which is from the North Atlantic, they've been seen feeding on alcyonasians.
Are we good
corals.
with this crinoid?
So
Watch lead.
it's not entirely unreasonable to posit that this critter
Okay.
is feeding on
Whenever you're ready.
the coral polyps
All right.
of these are Dipsodilophilia.
Thank you, Josh.
Yeah, that's
you
Lophelia.
want to push up and
Yeah,
push this
so
over.
that's not
Copy.
unreasonable to think that it's yet another
Yeah, just keep
corallivore
your heading that way.
feeding on these things.
Wow. And that would
So
explain why
yeah.
it's so numerous up here where there's so much live coral.
Sure. It could alternatively be feeding on the epizoites, on the dead coral, or something else that we're not seeing. But there's definitely at least some previously established observations that suggest these are predatory on
Yeah, we can hop
Cnidarians.
over to that feature.
Okay.
So that's another potential predator for this ecosystem, which may not have been observed before. for whatever reason.
Wow, interesting.
270.
Yeah,
thank you.
In addition to the other things that I'm seeing, the sea urchins, as well as that, although that Gilbertaster or whatever that goniasterid was, seems to be mainly interested in Plumarella, I guess. So
yeah, it sure looks like it.
Yeah.
310. I think it's actually where the.
But yeah, no, you're again. I'm a little surprised by
310.
all the things you're seeing on these dives, because a lot of this I thought was already known, and it's turning out to be quite exciting. So
awesome. Great. Thanks so much, Chris.
20 meters.
Thanks. Have a good night.
Okay, hopefully we'll hear from you again.
As long as I can.
Great. Yeah, I know. It's getting late.
Oh, it's not so bad, but I got stuff. Okay. Thanks. Bye.
Thank you.
How long do you think you've been flying for, Fernando? Don't look at the clock.
I don't know. An hour and 15 minutes. What time is it?
He looked at the clock.
No, I have no idea what time it is.
You've been flying exactly an hour and 15 minutes.
Oh, really?
Yeah.
Oh, wow.
Nice job.
Yeah, it's been an hour and 15 excruciating minutes. Okay, so...
Great job, Fernando.
You are doing a good job. This is tough flying.
Go ahead. So, like, if we turn, then the tether would be pushing into the vehicle, right?
Yeah, I mean, you can go over and look down between your toes.
Yeah.
And maybe turn a little bit to get a profile. I don't think we'll be able to get any real zooms on that side.
Yeah. Okay, I can try it.
So why don't you center up on me here?
Okay.
And then,
maybe like, down a little bit, forward, turn right, and then lateral to port to fight the current would be my guess.
So we're trying to see whether we can just get a peek over the far side, the south-facing slope, and just get a perspective that might be similar, would give us a similar view to what we've been looking at from the side that we faced,
if you want me to come out
that we climbed, descended earlier.
Copy. All right. Yeah, I think my laterals are 100% right now.
yeah. Yeah, do not turn your head. Come back to port.
Come back to port.
So all you can do is really look down.
All right.
So keep coming to port. See how your tether is
Yeah,
lined
you can
up.
see a lot more coral over on this side, as we would have expected.
That's a lot of current.
It would be interesting to see if the entire face is covered with coral.
Yeah.
To look at sort of the depth stratification, but unfortunately we can't do that today.
Can we peer into the darkness
We'll
here?
have to wait till a calmer day.
Yes.
Like.
No, I'm just asking if video can push it.
This gives us a good idea.
I mean, can you brighten it anymore? Or is
No,
it
this is all we got. I can go in and try to up the gain if we want.
Like they're in rows.
Yeah. So another unusual aspect about these Scleractinian coral mounds is that one genetic individual might have a life of thousands of years.
Yeah, that's about
because
all I got.
they can break off and continue growing. They can just fragment, and then that fragment
I think
will
that's as much
establish
as we
a
can
new
do on this side.
colony. But it's the same genetic individual, and it can basically wind up living that way for a long, long, long time compared to many animals.
Move complete. Thanks, Bridge.
Yeah, so watch Lee. He can't even turn around with this current. Looking at his thrusters, when he turns broadside, he's 100% on the thrusters. So. We can come back down and get the ridge, Fernando.
All right.
Turn your heading to port.
So that's our glimpse of the south side, south-facing slope of this feature.
I think we were lucky to get it.
We are lucky to get it.
Right.
Now we're going to tuck
now
back
you're
in for
heading
safety.
to the current. So that should make it a little easier for you.
About all they could handle with the thrusters, the pilot said.
What was the answer? Like half a knot of current down here?
Yeah.
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 We still moving?
Just completed.
Yeah, they
So
closed.
you might be moving just a little bit.
Closed down a little bit ago.
Yeah, we could probably keep that move going.
Is that like a different fish in the
I
center?
think it is.
Go ahead.
Your heading is.
There's something down there.
Oh, we've got a fish of some sort.
You want me to
Looks
take
like a
your
different
rotator?
one than we've seen
Yeah.
today.
Beautiful.
Sure. Well, I'm pretty high up, so I need to drop quite a bit.
Oh, good.
So let's
Ken
come
Sulak
out
is still
on
on the
video. Let
line.
me
All
drop
right.
because I'm way too high. Like we're looking down pretty much.
That's full wide.
Copy. Let me just bring it up a little bit. Okay.
So Josh, your heading's at 260.
All right, let's come in.
255. You still want 310 on the move, or do you want?
Yeah.
Yeah.
Yeah, because that
Right
310 is
The ridge.
the ridge, and
We
we're
wanna
trying to go across the ridge, right?
Yeah. I'm trying to ride it.
Go ahead. Right there.
We'll
Hold.
get another one underway. Bridge, this is RV nav.
Oh,
Yeah,
let's
I'd like
come
a
up.
move, same as last. Range 20 meters, bearing 310 degrees, speed decimal 2.
Pins are beautiful.
Yeah.
It's not going to make it easy on you.
Yeah.
So Ken Sulak was in the chat room reminding us about the difference between the Lophelia
All right.
mounds that we see here on the Blake Plateau versus those in the Gulf of Mexico. And in the Gulf, the mounds are actually built on salt domes.
Copy that.
So the actual live coral is probably higher in cover, but they it doesn't penetrate the surface or the seafloor. There's only a couple of meters worth of coral in any one place, not the hundreds of meters that we see in the Atlantic. Wow, that's a great view of that fish. I know I've seen him before, but I can't remember. An Oreo, I think Scott, Sandra Brooke, just and Scott France just
awesome job
commented that it was an Oreo.
Thank you.
Couldn't think of the name. Thank you.
I was thinking cookie
But anyway, so the Lophelia that we find in the Gulf of Mexico is quite a bit different in how it's formed and probably in the age compared to what we see in this area of the Southeast Atlantic or off the Southeast Atlantic coast.
Nice.
And Ken then turned around and identified the fish, agreed that it is an Oreo and Neositis species. It's a genus is the Neositis. We're seeing that this Limonema species is dominant around here. What did you call it? The coral hake?
Yep. The coral hake or the reef codling are two common
push
names.
Up or down. Don't be afraid to gesture Z-bias too.
Yeah, the thing is I have to push up and down all the time.
Well, then never
Right there you can see the stages of the living coral on the right, the bright white, sort of that light brown in the middle on which it's growing, and then below that is the long dead coral getting darker and darker, stratified by age. Be interesting just to see what the age of the base of that is. we might be able to get a sample at some point. We might want to consider doing that, just to see one of these smaller mounds to see what the oldest coral is at the top.
Yeah, that's true.
Top of the mound and within the smaller mounds.
It would be quite a precarious thing to sit down anywhere
That's
around
definitely
here.
the issue.
Star, yeah.
There's another sea star,
right.
leaving the view at the bottom
Looks
center.
like it's cut it down in.
Do a quick zoom of that little guy.
Try to.
If possible.
Yeah. Yeah, we're like right on top of it.
Come down a little bit.
It's times
You
like
can
this
go
you
and
wish
push
you
past
had
the skids. There you go.
a person
Already.
down
Move
there
complete.
to get a
Thanks,
sense of
Bridge.
scale. I don't know if one of these gullies would be as tall as a person or up to their ankle.
Yeah. I think it would probably be more like
Knee-high,
the
maybe.
former.
Yeah.
But,
the former? You think it would be more like a person high?
Oh, I would think so, but I guess it's hard to say when we're zoomed in
Yeah,
like this.
yeah. We'll get the lasers on.
Oh, and look, that sea star is on a plumerella
again. I think.
Go ahead.
3 for 3.
Why is such a small twig support
And
such
it
a
does
large?
look like the same species.
That's a good question.
It is kind of amazing that that very fragile, delicate-looking coral can hold up such a big sea star.
It is amazing.
So insightful, navigator.
Well, I think it's mostly water in there, so it's really not a lot of mass, but it just looks like it,
All right. Let's come out video.
All right.
That's great. Can we get the lasers on just for a minute to get a sense of scale here?
Yeah, they're on. You just have to tilt up.
That's gorgeous.
Oh, they are on. They're just so rugose you can't see them. Maybe in one of those flat sediment areas, right in the...
Yeah, it's gonna be tough. Yeah, no, it's gonna be impossible to get it.
Sorry.
Just too much coral. We just can't.
Yeah, just drop down here a little bit.
Can't see the lasers. We can't figure out how big everything is. Oh, there you see them a little bit.
I see one. I haven't seen two together. They're definitely on.
Oh, there they are, right there.
Yeah, but not together. Okay.
I should have.
Gonna keep on
I
pushing
guess
out.
that sea star was less than 10 centimeters because the lasers were on, but we couldn't see we couldn't see them in the view.
Oh, so going back to that cute little Oreo fish that we saw earlier, Ken Sulak said that Oreos and their close relatives are one of the few groups of fishes in the deep sea that actually rip polyps off of corals. Tip nippers.
Tip nippers.
Scott Brett said.
Say that 10 times fast.
Never mind.
Yeah, we don't want to. This is live.
Sounds dangerous.
Going.
Our chat room is slowly getting smaller as the end of the workday nears.
Yep. Only the diehards.
Looks like maybe a sponge.
Looks like one of the sponges there growing in the coral. The dead coral matrix.
You may have been seeing it earlier, but there's another one of those chimney sponges that we were looking at yesterday.
Mm-hmm. Yeah.
Oh, there are the lasers. Okay, so that one mound is a little more than a meter, maybe.
Mm-hmm.
Is that a crab? Just
And
to
that
the
looks
right
smaller than what we were looking at previously.
So
right
these
above
are
lasers
about,
now.
what, waist high?
I think that's
a little more than waist high. 3 feet, so. They're big. Definitely not ankle high.
It's There's a big coral
and going back to the mound formation in different parts of the Atlantic, Ken Tulak said that those in the eastern North Atlantic are formed like what we're seeing here, where you have
a little more north.
Extensive fields of rubble building to a very thick elevated mound.
Oh, that's a beautiful
wow, that one on the right is so alive. That's gorgeous.
Picked a good spot.
Bridge, this is ROV nav. Yeah, I'd like a move 20 meters, bearing 320 degrees, speed decimal 2.
Want me to take your rotator?
Sure.
It's like it's floating.
Yeah.
Good copy.
Another tail of something behind him.
Could we get the lasers off?
Oh, it's just one of the cutthroat eels, I guess.
Thank you.
So you're pretty high up. And
that's beautiful.
You probably come down a little bit.
Okay. Getting closer and coming down.
This edge
and from the genetic connectivity work that we've done on Lophelia, each of those regions, the Gulf of Mexico, the southeastern U.S. here, and the northeastern Atlantic are all genetically differentiated.
So you can, get low enough.
You can see a pencil urchin way in the background on the left. Sideros, or Siderus.
That we learned means some kind of crown, right?
Crown.
That's what Chris said. A special Persian crown.
There's
was
another
that
one in the lower right, just barely peeking into the screen.
It's interesting that piece of coral behind this very live one is exactly the opposite. Very dead.
Very dead.
Very covered in brown.
All right. That's full wide.
Copy.
Oh, and here's a whole bunch more of the, what we've been calling duvia on the left. I think Sandra thought that it might actually be something else besides duvia, so we have some samples, and I'm sure we will figure that out in the next upcoming months.
Going back.
Any idea what it might be? Is Sandra still on? She may not be.
Yeah, she may not be.
She is still on the chat room, but we haven't heard from her. Wow, that is a beautiful piece, isn't it?
Yeah. I think that's the biggest live coral head that we've seen.
You have your rotator back.
Copy.
You know,
on the right side of the screen. Where is it again? Okay.
The off color one to
your
left.
Copy.
looks like one of our, a different octocoral maybe that we're coming up on here.
You know, this face we're looking at, I'm wondering if maybe at some point that gave away, and it's an erosional scarp. You can see.
Hold right there.
Oh, it's actually Madrepora oculata, so that's actually a stony coral. It's just
not pink,
a paler, paler version.
Probably sit down.
Another crinoid.
Back up just
and there's a
bit.
crab over on the left.
That's
the golden crab variety, it looks like. Did you see it?
No, I didn't see it. I was staring at the coral.
Yeah.
There you go. Now it's still.
He's now hidden.
Okay, go ahead in.
All right.
Nice. You gotta put on your fog light.
Wow, it's a beautiful view. You can see the polyps out. Seem to be facing away from us.
And that's a partial.
Go ahead.
Look at that.
Nice.
Gorgeous shot.
Nicely done, Fernando.
That's
wow, looks like he has
framed.
a couple of barnacles, gooseneck barnacles.
We have more zoom?
And
several
a little
large ones.
That's full zoom.
In the center of the screen.
And there's a little crinoid, stock crinoid in the background there behind that larger barnacle.
Maybe not.
Well, what's the aperture set for?
Not sure.
What's the focus set for?
I don't know.
It's not very useful here. We're going to turn off the fog lights for a couple of these shots.
Move complete. Thanks, Bridge. Pretty sure it's set for like.
Pilot, there was a
foot from
crab
the
hiding
toes,
to the left, but I think we might have to be higher.
That close?
Okay, whenever you're good video, or do you want to more zoom in?
That light pool is pretty far.
Well, if the aperture is
I think I'm good if science is.
Okay.
This might be a good place to get the crinoid.
Okay.
All right. Let me rotate
Yeah,
to
yeah.
crinoid.
Quick, sorry.
Yeah, I'm not sure.
Go ahead. Yeah,
Yeah, he was settled
you
down.
can probably do it.
Put your toes
Yeah.
against the dead stuff.
we'll try watch
wonder
lead.
if it
Let's
would-
come in. Video.
Come down.
Might be a good idea to get
Are we moving?
crinoid if
Okay.
So we're going to
Maybe
settle down
settling
and
out.
take a
All
little,
right, that's
try
full
to
zoom?
take a sample
No, that's partial.
of this
Go ahead.
Madrepora. This
Let's
is
take
actually
this
one
shot
of
and
the
then I'll
Aspire
get set up.
target corals.
Alright.
We haven't taken any samples
Moving
today.
down
We have
slowly, trying
almost an
to-
hour left on the bottom, and we have actually found a flat spot to sit the ROV down, so there's a chance we might be able to get our first sample of the day here.
So we don't have any samples, right?
That's correct. Yes, a lot of open boxes.
All right, video whenever you're good.
Wonder if they want to do.
Alright.
That looks nice.
Yeah, you're doing a great job with this current.
All right, whenever you're good.
Alright.
Okay.
All right, so you're going to have to get closer to this.
Yep.
So maybe push up a little more to your starboard, and then you're going to have to use auto heading.
he's still hidden quite well.
So watch your left toe there.
Yeah, you can just see a crab peeking out on the left under that
gotta
other
get,
white coral.
Closer.
We passed
Yeah, so
over him
keep
earlier.
turning.
He's tucked away pretty well.
And then come forward and down.
It's another of the golden crabs.
A little more to your starboard.
Looks like a
Still too far out.
Laminema millinarum on the left.
Full wide on the HD.
Right behind a crinoid. There's a lot of crinoids here.
Is that good?
You might be able to reach that.
It's like an
You turn your
echinoderm
heading
shell
to port
over
more.
on the right on the sediment area, and there's a living form of it above it.
Right there. I'm afraid of hitting the crinoid.
You're still pretty far from the crinoid.
Okay.
Yeah, right there.
All right.
So you're going to want to put in auto heading.
You want me to punch it for you? Oh, yeah, nice.
All right, and now
The
drill
crab
lock
is
once you're settled.
peeking out a little bit still. I see his leg moving.
That's pretty good.
All right. Can somebody punch it in for me?
Got it. Boom. And then
Drill lock's in. All right, hydraulics are up.
Copy.
Yeah, you're stable.
All right. We're not moving. Everything's open, right?
Everything's ready to go.
Okay.
You want to keep an eye on Sirius?
absolutely.
Nav. So this is going to probably be brittle, I'm guessing.
Okay. So, what is it? How do I check the comms or
No,
need?
you don't
Okay.
have to check the comps.
All right.
So once you turn it on, then you can see if force feedback is on or not, and your grip force.
Okay.
Once you hit the.
So enabling.
Just be careful, you're on the edge of this table here.
Okay.
You don't want that to fall off. You might want to slide your chair forward.
But since it doesn't have a lock. Okay.
Right.
And then grip lock. So grip locked, and then grip force.
What do you grip lock on?
So the grip lock is the same as the left button there. So you hit force feedback's on, which is great. Grip force 5.
5's a good one.
Yeah, I think you're set. You're ready to go.
Okay. So.
Yep, there you go.
All right.
You're live.
Be wary of that index button.
Yep. So this is going to be brittle.
Okay.
So you're probably going to want to maybe take some off the top or off the bottom edge here, whichever you feel comfortable with. Practice closing your jaws before you get up there.
Okay. Can you zoom in on the main HD video a little bit?
This is our first sample of the day. We've been on such a steep slope and such strong current. We really have not had the opportunity to just sit down and relax for a minute. Although I wouldn't say the pilot is relaxing. So we're going to get a sniff of this madrepora in front of us. So that its DNA can be analyzed.
Sometimes you can like bump it with just your finger to give you an idea of what it's going to be like.
Okay, let me close the jaw.
And if there's time before we have to move on, we may get a snip of the Lophelia.
Is this going to be a brittle coral?
Yes, definitely brittle.
So in this case, I have to. All right, let me line it up with this right here. So should I bring it down like this and just clip like this?
It's not really going to clip. It's just going to break as you bring the jaws together. So you might try to there's two options. You come in like you're clipping it and try to squeeze some within the Tygon, or you can try to get some between your fingers.
So it's either a cradle situation or a Tygon.
Okay, let's try the second one. I mean the first one. So I'm gonna bring it out. Go a little bit more horizontal.
If you're going to try to snip, you might want to go more towards the back.
Okay. So let me just bring up the camera.
Yeah, Bobby's suggesting maybe scoop was also an option.
Let's try this. Can we come in on Mini Zoos?
Yep.
Video.
It's hard to see the geometry of this guy, you know?
Right there, that's good right there. So, indexing out. Oh, moving.
Moving back a little bit.
Just saw cutthroat eels swim by. We've seen a lot of them.
Hold on.
Not as many up here on the top, though. They seem to prefer the lower, deeper areas.
Okay, I'm not moving anymore.
It's a crinoid on the upper left.
Okay, indexed in. Can we move the Mini Zoos to the top of it again?
I'm going to try to get a sample of crinoid.
A little bit to the left?
Time.
Yeah. Yeah, there you go, right there. I think.
So, see how you're already in coral?
Yeah, I wanted to get
very brittle.
It's extremely brittle. I wanted to just nip it right there.
You're going to have to bring the arm up a little bit. push forward.
Okay. Yeah, I think you're
fully extended there.
All right. So do you need to bring it back a little bit and then rotate to the left? Okay.
I think you might need to move the whole vehicle forward a little bit too. Would help you. Reset the vehicle down maybe. But
all right.
Don't move it now. If you're gonna move the vehicle, you got to move the arm out of there.
Okay. Right there is good. Or do you need to stow it?
That's fine.
Okay.
Should I come wide on the cameras?
Sure. On this one right here. Thank you.
Yep.
Yeah, remember you're in auto heading.
Oh,
you're moving the set
okay.
point. Can we come out on Mini Zeus?
Yep. Full wide.
I think the current might have increased because I'm going. I'm not going forward.
What are you trying to do here, Fernando?
I was trying to not drift back, but I'm not moving forward at all.
Get a little bit of forward way left. You're at 50%.
But this is full deflection, though. Is it because of the arm, or?
Like a heading change.
Hold on. So let me just land and stow the arm.
So we're getting a good view of a very steep, knife-edged portion of the ridge.
So try and face faces
and it really looks to me like an erosional scarp that has had some overgrowth.
Okay, until now I can't turn.
We're experiencing a lot of current here.
No, I didn't see any faults.
Okay.
So let me just
so far
right
unable
here
to get that sample.
Okay. So right there. Yeah, watch lead.
Do you want me to grab that sample for you?
Go back? Yeah, if you want.
I can get it.
Do you want me to stow the arm? Or that's
leave
fine.
it where it is right now.
All right.
We're having a quick pilot change.
So Josh, I think what he was saying was he had the stick all the way forward and he wasn't getting 100% on his axials. So take a look at that.
See a piece of bamboo coral right in the foreground. Several of the Anthomastus, the red star-like soft corals. climbing up that steep slope again. Another crab, that might be the same one. We finally chased him away.
Hey, where's that coral?
I think it was to the right.
Starboard. Can you turn your heading?
Gotta find that madrepora again, see if we can do that. Pilot, the one on the left, the pinkish one, is the one we're looking for.
What was that watch lead? You're very soft.
Sorry, pilot, the pinkish one on the left.
You want the little one, this different one over here? Is that the sample you want?
The pinkish one, yeah. Right
This
in the
one,
foreground.
center.
Center.
All right, this is different than the one we were grabbing, just FYI.
That's the species we're looking for, yeah.
All right, you got it.
Cheryl's gone to dinner, so anyone in the chat room, correct me, but I'm pretty sure that's the same thing. The madrepora.
The actual colony we were going for before
is on the starboard.
Yeah.
Yep. All right, let's zoom in on this real quick.
All right. It's a partial.
All right, let's come on out.
Full wide.
I'm going to end up grabbing this whole colony here. If you can extend the draw, go for it.
Copy.
Yep, it's clear. All right, stop there. Stop there. All right, port inner.
Pilot.
Yes.
If you could also get one of those broken stalks of the Lophelia from on the lower left that just broke off. Yes, that was another sample, and that would go in a different bio box, please.
All right. So any one of these white pieces would be good?
Well, one of them was just knocked by the skid, so on the lower left, might as well just see if you can get one of those long stems was broken.
Okay, go ahead.